two manner. Either it can act as an extracellular messenger, or it can also act as an intracellular signal. Okay, we will be discussing on its importance as an intracellular signal. Okay, but it can also act as an extracellular messenger. Okay, so as I mentioned, it can act as an extracellular messenger or as an intracellular signal. Now, when I'm talking about intracellular communication, okay, just in the way that we discussed about CAMP, calcium, and all, wherein we referred them as second messenger, same way your nitric oxide can also be referred as a second messenger. Okay, so nitric oxide plays an important role as a second messenger and helps in intracellular communication. Okay, now let's understand how it is produced, and how it is related to its function. Okay, so nitric oxide is produced from an amino acid basically. Okay, so you might be knowing the amino acid called as L-arginine. Okay, L form of arginine. So arginine is the amino acid being utilized here as a precursor for synthesis of nitric oxide. So nitric oxide is synthesized from L-arginine, and this reaction will require oxygen and NADPH. Only then the reaction will happen, and this is catalyzed by an enzyme called as nitric oxide synthase. Okay, so it's a synthase enzyme. By the name you understand, the enzyme is responsible for helping in synthesis of some compound. So here it is nitric oxide synthase. So by the name, it is capable of generating nitric oxide. The substrate here is arginine. Using arginine in the presence of oxygen and NADPH, the reaction is being carried out, and you have production of nitric oxide being taking place. Now, nitric oxide has got certain functions in the cell, as in it is going to play an important role in anticoagulation process, neurotransmission, smooth muscle relaxation, and also visual perception. So these are some of the areas where these nitric oxide plays an important role. Okay, that is anticoagulation process, neurotransmission, smooth muscle cell relaxation, and visual perception. All these are some of the important functions attributed to nitric oxide. Okay, now this discovery that happened long back, okay, somewhere in 1970s the studies began, and somewhere in 1980s to 1986 things were clear that nitric oxide plays a very important role. So long back from those days when the studies were going, it was by an accidental observation that the scientists could understand that nitric oxide plays an important role as a messenger molecule. So his intention was not to understand NO as a second messenger, okay, or as a messenger molecule, but he studied to understand that how exactly the function is happening, smooth muscle cell relaxation, how it is happening. To understand that, he was studying the same, but during his study, he accidentally observed that they play an important role as a second messenger. Just a minute. Yeah, so as I was telling, that nitric oxide, they functions as a messenger molecule, and it was accidentally observed, okay, by the scientists when they were studying on how exactly parts of aorta, heart aorta, that is a blood vessel, okay, they were studying with heart aorta, and while studying that heart aorta, they accidentally observed that nitric oxide is a actual second messenger molecule which is involved in smooth muscle cell relaxation. So that is how they discovered that nitric oxide functions as a second messenger. So let's understand that how that experimentation was conducted and how could they understand that this is one of the important second messenger. So the study was all concentrated upon a neurotransmitter called as acetylcholine, which we have already discussed in several examples. If you'll recollect, in phosphoinositide topic, I had discussed about importance of acetylcholine, where it binds to as an extracellular messenger and brings about muscle cell relaxation, and also we're involved in secretion from mast cell for production of histamines. The same way here also we have taken acetylcholine as an example here because the scientists were studying those at that time. Okay, so acetylcholine, how exactly it caused a relaxation of smooth muscle cells of the blood vessels. So it was in vivo, it was proved that acetylcholine is a neurotransmitter and it helps in causing relaxation of smooth muscle cells. But the same they wanted to study under in vitro condition to understand how the mechanism is, how exactly it is actually happening. To understand that, they tried to do the same under in vitro condition. So when under in vitro conditions, several scientists who were working with this, they initially started working with portions of that major blood vessel, that is the aorta, which I already told sometime before, that the one of the important blood vessels to your heart is aorta. So that aorta they worked with, they took some tissue of that aorta, okay, and they incubated it in some concentration of acetylcholine. That is a physiological concentration. That means at the natural, naturally, whatever is the concentration of acetylcholine associated with our cells, at that same concentration, they provided acetylcholine and they checked out what is the response of those particular aorta tissue under in vitro conditions. But they didn't get a good response. Rather, certain times they didn't get a response at all, and certain times the response was very little. That means the response was very, very less. Okay, so they couldn't get a good intensified activity there. So they understood that they were not really doing well with this particular study. So somewhere in 1970, okay, one scientist by the name Robert Fugard, who is a pharmacologist basically by profession, okay, from New York State Medical Center, he also tried studying the same, okay, but in his particular process of studying, he was actually studying with the rabbit aorta, and he was trying with various agents apart from acetylcholine also. So he was studying acetylcholine as well as other agents also in his study. So somewhere in the study when he was doing this, okay, he initially studied using strips of aorta. Strips of aorta matlab, the blood vessel aorta, from that some tissue were just collected, okay, in the form of a small strip. It was cut out, and that particular strips were utilized, okay, for the study. So he just dissected that organ, and from that a small tissue strip was taken off, and that was incubated into acetylcholine, okay, and he checked how the response was happening. But again, he was not getting a good kind of response. But due to some technical difficulties, okay, some technical issues that he was facing, he's tried to switch over to aorta rings. Okay, so the blood vessel ring was taken. So he could take a horizontal section of that particular blood vessel, and a small ring was taken. Okay, so initially he was studying with a strip, but due to technical difficulties, he started taking a ring. Now, when he was studying with the ring, okay, the actual preparation responded to acetylcholine. Okay, so initially when he was studying with a strip, he saw that the effect was very little, or it was not much happening, or rather it was nil. There was no response. Okay, see, I'm talking about the blood vessel and the smooth muscle cell. Both I'm talking about. Blood vessel, when I'm talking about, it is made up of endothelial cells, and just below the endothelial cells are your smooth muscle cells, okay, which causes relaxation and contraction of the blood vessels. Okay, so those muscle cells I'm talking about. So he wanted to know that how this particular smooth muscle cells relaxed, okay, when acetylcholine was provided. So to study that, he was doing this experiment. So in the experiment, what he did was he took strips, small sections, okay, he dissected from the organ, from the aorta, and that tissues were incubated into this acetylcholine. And when it was incubated into acetylcholine, he was not getting a good response at all. So, but unknowingly, due to some technical issues, he actually switched over to another particular, fragment, that is a aorta ring. Okay, so he cut it in a horizontal fashion, the blood vessel. Okay, and the aorta ring was taken off, and that aorta ring was then introduced into acetylcholine solution. This time, he got smooth muscle cell relaxation. Okay, so by this particular experimentation, he understood that the complete layer, okay, of the particular aorta ring was required. Why? Because the problem where he was actually failing in his experiment was when he was taking a strip that he was cutting it off from the organ. During dissection, many a times what happens is if you dissect it in the form of a strip, there is a chance that you can, the upper layer, which is the endothelial, which is delicate endothelial layer, that might get lost certain times. Okay, so because it is a mechanical event, you are actually putting a mechanical tension on the tissue. You are cutting it off with some kind of a tool. So when that cutting is happening, there is a chance, that probable chance, that the delicate endothelial cell layers can be affected. So basically, because of that, the strips were not giving a good response. Okay, so when he tried out with aorta rings, he was getting response, because in aorta ring, he was not actually causing any damage to the endothelial layer. He was just cutting out a small ring, and the ring was taken up for the study. So that is where the endothelial cells lining that particular ring were unaffected, and he was able to get the response. So this suggested that endothelial cells were the actual one, they were involved in the response, okay, because if endothelial layer was not present, he was not getting response. When endothelial cells were present, he was getting response. So this made him understand, made him realize that endothelial cells were the ones which were actually involved in the response, and they were actually providing some sort of signal to the muscle cells so that the muscle cells can relax and your blood cells, blood vessels can get dilated. So this particular process of blood vessel dilation, okay, which is related to muscle cell relaxation, was caused by acetylcholine, but it was involved through, the process was happening through your endothelial cells. So Furchgott, the scientist Furchgott, he proved this, okay, accidentally, because he was not in an intention to do it with an aorta ring, but since due to technical issues, he actually switched over to aortic ring. But when he was using ring, he got this particular effect. So this gave him the understanding that endothelial cells were the actual ones which are involved in the process. Okay, they were the ones which are facilitating the transmission of signal to the adjacent muscle cells. In subsequent studies, okay, it was found that acetylcholine, it binds to receptors on the surface of endothelial cells, leading to production and release of an agent which diffuses through the cell's plasma membrane and causes muscle cells to relax. So after his study, so his study only just proved that it is endothelial cells which are involved in the action, but then the scientific community further worked on it, and then they understood that acetylcholine, where it actually binds is, it binds on the endothelial cell only, but on the endothelial cell surface, there are certain kind of receptors, okay, which are specific for acetylcholine. So when acetylcholine is released, the acetylcholine specifically binds to the receptor which is present on the endothelial cell. When it binds to the endothelial cell, the receptor gets active, and the receptor internally causes production, okay, of agent. That agent is nothing but your nitric oxide. So it internally helps in generation of nitric oxide and release of that nitric oxide, okay, which then is going to shift its location from the endothelial cell, and it diffuses and enters into your muscle cells. Once it reaches your muscle cells, it helps in causing relaxation of the muscle cells. So how exactly it is working? I repeat once again, when the later studies, they could prove that endothelial cells only were the one which was responsible for facilitating signal transmission to smooth muscle cell. But how exactly it happened? Acetylcholine had a receptor. Okay, so the receptor for acetylcholine was present on endothelial cell surface, that is cell membrane. Okay, so when acetylcholine binds to that receptor, the receptor gets activated, and the receptor internally caused, okay, transmission of signal, okay, to systems which will create product or create nitric oxide or produce nitric oxide, and the produced nitric oxide then leaves the endothelial cell, okay, and it diffuses into the muscle cell. Once it diffuses into the muscle cell, it causes relaxation there. So the relaxation is actually brought about by nitric oxide, and it is helped out by none other than your acetylcholine receptor internally through a signaling process. Okay, so the diffusible agent, okay, at that time was not known. It was only known that something is going from the endothelial cell into the smooth muscle cell, and then the relaxation was happening. So in 1986, okay, the diffusible agent was identified, and it was identified by Mr. Luis Ignaro at UCLA and Salvador Moncada at the Wellcome Research Labs, which is in England. Okay, so the scientist Luis Ignaro was responsible for identifying this chemical compound, and it was identified as nitric oxide. So let's now understand the mechanism behind this particular process, that how exactly acetylcholine brings about this particular effect. So let me show you a picture. I believe everyone is able to see the picture. Okay, just look at my cursor and try to understand as in when I'm discussing on it. Okay, so just see this. There are two layers of cells that you see here. Okay, one is the upper layer having a portion of the, you can see that 3 cells are actually there. The one part of the cell here, then a second cell here, and then a third cell at the top. So there are 3 cells at the top. Okay, one central cell showing all that mechanism, and beneath that you can see some other 2 type of differentially shaped cells, leaf shaped cells. They are nothing but your smooth muscle cells. So the upper cells are nothing but your endothelial cells, and the lower cells are nothing but your smooth muscle cells. Okay, so these upper three cells, what you see is nothing but endothelial cell. The beneath that you see some smooth muscle cells. So together they form your blood vessel. Okay, so this muscle cell is the one which is going to bring about relaxation and contraction, due to which your blood vessels can get dilated. Okay, so let's see, understand how exactly it happens. So as I already told, the extracellular ligand here is your acetylcholine. This is the actual fellow who brings about the response. So whenever the blood vessels need to be dilated, okay, at that very moment acetylcholine gets released as an extracellular messenger. Now this acetylcholine pairs up or binds with a receptor which is present on the cell membrane of endothelial cell. So endothelial cell has got the membrane, and the membrane has got a green colored receptor that you can see here. So on to that receptor, the acetylcholine comes and binds to it. Okay, so this is the very first step involved here. Okay, the very first step is acetylcholine coming and binding to the receptor. Once it binds to the receptor gets activated. Okay, the receptor gets activated. The activated receptor then can bring about activation of calcium influx process. Okay, I believe all of you know influx and efflux. Influx means something coming inside, efflux means something going outside. Okay, so it brings about a calcium influx. So it is activating certain calcium channel, which brings about an increase in the calcium. Okay, so that is the second step. So second step is nothing but elevation or increased concentration of calcium following activation of calcium ion channels by your acetylcholine receptor. Okay, the first acetylcholine binds to the receptor gets activated. That is the first step. Second step, acetylcholine receptor activates calcium ion channel, due to which lot of calcium starts coming inside the cytoplasm of the cell. Okay, that is the second point. Third point, whatever calcium that came across in the cytoplasm, okay, now it is an increased quantity of calcium, and I already told that calcium signaling always happens by shooting up the concentration of calcium inside the cytosol. So here increased levels of calcium will now activate an enzyme called as nitric oxide synthase. Okay, so increased calcium will now, using calmodulin, it can go and bind to nitric oxide synthase, and it will activate that nitric oxide synthase. Nitric oxide synthase, just some time before I told, this enzyme actually utilizes arginine, okay, in the presence of oxygen and NADPH, and helps in generation of nitric oxide gas. So nitric oxide synthase, in the third step, what it is doing, the activated nitric oxide synthase will attack arginine, and from that it will cause release of nitric oxide gas, okay, utilizing oxygen and NADPH in the reaction. So this was the third reaction, okay, or third step basically. In the fourth step, okay, what happens is the nitric oxide, which is now synthesized, okay, will leave the endothelial cell, okay, and will diffuse inside the smooth muscle cell. So nitric oxide will, whatever is generated here, will now start, okay, going out into the extracellular space, and from the extracellular space into the cell of your smooth muscle cell, into the smooth muscle cell. So there is a diffusion that is happening here, and the nitric oxide starts entering into the nearby smooth muscle cells. Here it is shown into two of the smooth muscle cells, one of the molecule entering this muscle cell, another entering the second muscle cell, okay, whichever it is. So once the nitric oxide enters the smooth muscle cell, okay, it is going to activate another enzyme called as guanylyl cyclase. Okay, nitric oxide, once it enters your smooth muscle cell, it is going to activate an enzyme called as guanylyl cyclase. Now, what is the role of this enzyme? This enzyme is capable of producing a compound called as CGMP. It happens from production, I mean the substrate for this is your GTP. So guanylyl cyclase enzyme helps in production of CGMP from GTP. Okay, the way you had adenylyl cyclase when we discussed GPCR, I told that there is an adenylyl cyclase which causes generation of CAMP from ATP. Same way, guanylyl cyclase will help in production of CGMP from GTP. That is the fifth step. So in the fifth step, there is production of CGMP. In the sixth step, CGMP then activates, okay, a protein kinase. Okay, CGMP then activates a protein kinase which can phosphorylate certain substrates that will help in relaxation of the smooth muscle cell. Okay, so CGMP then what does it do? CGMP will now activate a protein kinase, and that protein kinase, it will phosphorylate certain kinds of substrates which are involved in helping in relaxation of the smooth muscle cell. So this is how, okay, acetylcholine that binds outside at the surface of an endothelial cell brings about relaxation of the smooth muscle cell, okay, by production of CGMP, okay, involving nitric oxide as an important second messenger, which is actually helping in activation of guanylyl cyclase and thereby production of CGMP. Okay, so this is how your this is the mechanism behind your working of nitric oxide in causing relaxation of smooth muscle cells. Okay, so once again I will repeat the entire mechanism for you. Okay, I want all of you to listen carefully to this. Okay, so nitric oxide as a second messenger. Okay, how it is exactly playing a role. So nitric oxide, this nitric oxide is a gas here, okay, and acetylcholine, the extracellular messenger, it initially acts as an extracellular messenger. It comes and binds to the receptor for acetylcholine, which is present on the membrane of the endothelial cell. Once it pairs it with that particular receptor, it gets activated, and once it is activated, it can activate the calcium influx ion channel, which will help in introducing calcium into the cytosol at a high concentration. The high concentration of calcium that has come inside will now activate the nitric oxide synthase enzyme. It will activate the nitric oxide synthase enzyme, which will help in production of nitric oxide from the amino acid called as arginine, okay, utilizing oxygen and NADPH in the reaction. And the nitric oxide then so formed will leave the endothelial cell and enter the smooth muscle cell by diffusion. Once it enters the smooth muscle cell, it can activate guanylyl cyclase enzyme, which will convert GTP to CGMP, and the CGMP then activates protein kinases, okay, which can phosphorylate certain kind of substrates which are involved in causing relaxation of the smooth muscle cells. So this is how your system works. Okay, I believe this is understood. The mechanism is clear with you all. In case if anybody has not understood, please let me know. Please unmute yourself and let me know so that I can repeat.